Hormone synthesis refers to the process by which the body produces hormones. Hormones are chemical messengers that regulate various physiological processes and maintain homeostasis within the body. Hormone synthesis typically occurs in specialized cells or glands known as endocrine glands. Here's a detailed explanation of the synthesis process. Endocrine glands. Hormones are primarily synthesized and secreted by endocrine glands such as the pituitary gland, thyroid gland, adrenal glands, pancreas, and gonads, testes and ovaries. Each gland specializes in producing specific hormones that regulate different bodily functions. Stimulus for synthesis. Hormone synthesis is often initiated in response to specific stimuli, such as changes in blood levels of certain substances, nervous system signals, or other hormones. For example, the hypothalamus, a region of the brain, releases hormones that stimulate or inhibit the secretion of hormones from the pituitary gland. Transcription and translation. Inside endocrine gland cells, the process of hormone synthesis begins with the transcription of DNA into messenger RNA, mRNA, in the cell nucleus. This mRNA carries the genetic information necessary for synthesizing the specific hormone. The mRNA then moves from the nucleus to the cytoplasm, where it directs the synthesis of the hormone through a process called translation. Post-translational modifications. After translation, the newly synthesized hormone may undergo post-translational modifications to become biologically active. These modifications can include the addition or removal of specific chemical groups, cleavage of inactive precursor molecules, or the formation of disulfide bonds. These alterations are essential for the hormone to function properly and interact with target cells or tissues. Packaging and secretion. Once synthesized and modified, hormones are often packaged into vesicles within the endocrine gland cells. These vesicles may accumulate in the cytoplasm until a signal triggers their release. Upon receiving appropriate signals, the endocrine gland cells release the hormone into the bloodstream through a process called secretion. From the bloodstream, hormones travel to target cells or tissues throughout the body, where they exert their physiological effects. Feedback regulation. Hormone synthesis and secretion are tightly regulated through feedback mechanisms to maintain hormonal balance and homeostasis. Negative feedback loops help prevent excessive hormone production by inhibiting further synthesis and secretion when hormone levels reach a certain threshold. Positive feedback loops, on the other hand, amplify hormonal responses to stimuli, leading to increased synthesis and secretion until the desired physiological outcome is achieved. Transport and metabolism. Once hormones are released into the bloodstream, they circulate throughout the body, binding to specific receptors on target cells or tissues. Hormones may also undergo metabolism or degradation in various organs, such as the liver or kidneys, to facilitate their elimination from the body. In summary, hormone synthesis is a complex and highly regulated process involving transcription, translation, post-translational modifications, packaging, secretion, and feedback regulation. This process ensures that hormones are produced and released in response to physiological demands, maintaining proper bodily functions and overall health. Hormones are molecules synthesized by endocrine glands that regulate various physiological processes in the body. While the synthesis of hormones occurs primarily in specific glands or cells, some hormones are stored within these glands or other tissues before being released into the bloodstream. Here's a detailed explanation of the storage of hormones. Storage organs and tissues. Hormones are stored in specialized cells, tissues, or organs within the body. Endocrine glands, such as the thyroid gland, adrenal glands, and pituitary gland, often serve as storage sites for hormones. Additionally, certain hormones may be stored in specific tissues or structures, such as the liver, pancreas, or adipose, fat, tissue. Storage forms. Hormones can be stored in various forms depending on the specific hormone and its properties. Some hormones are stored as inactive precursor molecules or prohormones, which are converted into active hormones when needed. For example, thyroxine, T4, the primary hormone produced by the thyroid gland, is stored in the thyroid follicles as an inactive precursor until it is converted into the active form, triiodothyronine, T3, and released into the bloodstream. Intracellular storage. Within endocrine gland cells, hormones may be stored in specialized vesicles or granules until they are needed for secretion. These vesicles protect the hormones from degradation and allow for rapid release in response to physiological signals. For instance, the adrenal medulla stores catecholamines such as epinephrine and norepinephrine in chromaffin granules until they are released into the bloodstream during times of stress or arousal. Extracellular storage. Some hormones are stored outside of endocrine gland cells in the extracellular matrix or interstitial fluid. For example, insulin-like growth factors, IGFs, are stored in the extracellular matrix of various tissues, where they interact with cell surface receptors to regulate cell growth, proliferation, and metabolism. Regulation of storage. The storage of hormones is tightly regulated to maintain hormonal balance and respond appropriately to physiological demands. Hormone storage is influenced by factors such as hormonal secretion rates, receptor signaling, feedback mechanisms, and cellular uptake and release processes. For instance, the release of stored hormones from storage vesicles or tissues is triggered by specific stimuli, such as neural signals, hormonal cues, or changes in blood concentrations of certain substances. Dynamic storage. Hormone storage is a dynamic process that can be modulated in response to various factors, including hormonal signals, nutritional status, stress, and environmental cues. For example, the storage of insulin in pancreatic beta cells can be influenced by glucose levels in the bloodstream, with high glucose levels triggering increased insulin synthesis and storage. In summary, hormones are stored in specialized cells, tissues, or organs within the body in various forms, including inactive precursors or active molecules. The storage of hormones is a dynamic process regulated by multiple factors to ensure proper hormonal balance and physiological responses to internal and external stimuli. Hormone secretion is the process by which hormones are released from endocrine glands or specialized cells into the bloodstream or extracellular fluid. This release allows hormones to travel to target tissues or organs throughout the body, where they exert their physiological effects. Here's a detailed explanation of the secretion of hormones. Stimulus for secretion. Hormone secretion is typically triggered by specific physiological signals or stimuli. These stimuli may include changes in blood levels of certain substances, neural signals from the nervous system, hormonal cues from other glands, or environmental factors. The secretion of hormones is tightly regulated to maintain homeostasis and respond to the body's needs. Synthesis and storage. Before secretion, hormones are synthesized and often stored within endocrine glands or specialized cells. Hormones may be synthesized as inactive precursor molecules or prohormones, which are converted into active hormones when needed. Additionally, some hormones are stored in vesicles or granules within endocrine gland cells until they are ready for release. Secretory mechanisms. Hormone secretion can occur through various mechanisms depending on the specific gland or cell type. In many cases, hormone secretion is regulated by a process called exocytosis, in which hormone-containing vesicles fuse with the cell membrane, releasing their contents into the extracellular space. This process is often triggered by specific signaling pathways or stimuli that induce changes in intracellular calcium levels, leading to vesicle fusion and hormone release. Bloodstream transport. Once released into the extracellular space, hormones enter the bloodstream or nearby lymphatic vessels, allowing them to be transported throughout the body. Hormones can travel significant distances in the bloodstream and reach target tissues or organs located far from the site of secretion. Binding and action. In the bloodstream, hormones bind to specific receptors on target cells or tissues, initiating signaling cascades that lead to physiological responses. Hormone receptor binding triggers various intracellular processes, such as changes in gene expression, enzyme activity, ion transport, or protein synthesis, depending on the hormone and its target cells. Feedback regulation. Hormone secretion is often regulated by feedback mechanisms that help maintain hormonal balance and homeostasis. Negative feedback loops are common in hormone regulation, where rising hormone levels inhibit further hormone synthesis or secretion, helping to prevent excessive hormonal effects. Positive feedback loops may also occur in certain situations, amplifying hormonal responses to stimuli until a specific physiological outcome is achieved. Temporal patterns. Hormone secretion can exhibit temporal patterns, with hormones being released in response to specific daily rhythms or circadian cycles. For example, hormones such as cortisol, growth hormone, and melatonin follow circadian rhythms, with peak secretion occurring at specific times of the day or night. In summary, hormone secretion is a highly regulated process involving the synthesis, storage, release, and transport of hormones throughout the body. This process allows hormones to exert their effects on target tissues or organs, coordinating various physiological processes and maintaining overall homeostasis.